Hi everyone, I am Saniya Kuller, and this little cutie is cute little Yankee. He's blowing you guys kisses. And in this video, we're going to learn how you can use technology like R to generate a sequence logo for a DNA motif model. So, this is kind of continuing the sequence of videos where we're talking about how you can use a PWM. I know I call it a probability weighted matrix, but it's actually called the position weight matrix, which gives the probability of having a given DNA base, for instance, in a given position of the motif. So we're going to see how we can use that probability weighted matrix and then end up creating a sequence logo using the seqLogo package in R. So are you ready? And again, please like, subscribe, and share this video with others. And please reach out with any questions that you have. That's what Yankee's seeing. So I'll be talking about a few different examples here. So basically, again, we're looking at how we can generate a sequence logo for a DNA motif model. So we have information from a position weight matrix, so the probability of a given base in a given position of the motif. That's what we'll be looking at. And again, I do talk in another video about how we can generate a sequence logo just from motif occurrences. If you have a set of sequences where that are occurrences or examples of a motif occurring, how can you get the sequence logo? So here we'll be talking about how you can get the sequence logo from a probability or a position weighted matrix. And I'll be going through some examples that we've kind of looked at before. So in a previous video, when I was talking about expectation maximization, I talked about how we have a set of DNA sequences, 4 DNA sequences, each that are 6 bases long, so they have to be the same length. And we were looking for motifs of with 3 bases here. We looked at mean, the multiple expectation maximizations for motif elicitation. So we ran this algorithm, and I have very detailed videos, so please do see the bio. But we talked about how we can run mean, multiple expectation maximization. So basically, we have the set of 4 DNA sequences, and we say, hey, please learn motifs of with 3 bases. And then what we ended up finding is that the optimal motif was here. The optimal was here, CAG, there was a GAG, and CAG. This was the occurrences of the motif that our model predicted. But apart from talking about those, and I talk in another video about how we can use CAG, GAG, and CAG and get a sequence logo from that. But here, I want to talk about the probability or the position weighted matrix that we get. We got in the background, and we had these motif positions here too. So what these tell us is the probability of having a given DNA base in a particular position. So here, these are the backgrounds. So the probability of having A in a non-motif position is 20.3%. C in a non-motif position is 23.1%. G in a non-motif position is 32.6%. And T in a non-motif position is 24%. But we're not going to be concerned about that when we're trying to do the sequence logo. We don't worry about this part here. We focus only on the motif parts. So this is the probability of a given base being in the first position of the motif, because our motif width was 3 bases. So for instance, 16.1% probability of an A in the first position of the motif, 37.3% chance of C in the first position of the motif, 33.7% chance of G in the first position of the motif, and 12.9% chance of T in the first position of the motif. And similarly, this is the second position of the motif. So 53.3% chance of an A in the second position of the motif, 15.5% chance of having a C in the second position of the motif, 17.8% chance of G in the third position, and 13.4% chance of T in the second position of the motif. And lastly, this is the third and last position of the motif. So the probability weighted matrix always goes from k equals 1, or the position weighted matrix to k equals w. So w is 3 here, the width. So again, 15% chance of A in the last position of the motif, 13.4% chance of C being in the last position of the motif, and 58.4% chance of G being in the last position of the motif, and 13.1% chance of T being in the last position of the motif. So we learned the probabilities of having a given DNA base, this A, C, G, or T. These are the only 4 DNA bases that we have. So the probabilities of them being in a particular position of the motif. And all of these need to sum up to 100%. 100% going across. That's how to interpret what they are here. So basically, what we had gotten then is we learned this final position weighted matrix here. So again, I just got rid of the background because we only cared about a given position of our motif. So again, K could be 1, 2, or 3 because our motif width is 3 here. So from this final position weighted matrix here, so we expect kind of like C being a bit more, A being the highest here, and G being the highest here, corresponding to C, A, and G. How do we take this matrix and represent it as a sequence logo? So here's a spoiler alert. This is the sequence logo that I got using this position weighted matrix. But I want you to see how we got this. So the y-axis is the information content in bits, and the x-axis are position. So k equals 1 basically maps to k equals 1 maps here. Then k equals 2. The second position maps here. And the third position maps here. So what you'll see is kind of the base at the top is either the base that has the highest probability or it's kind of tied with another base. That could be the case. And we'll see an example where 2 bases have like the same probability here. For instance, multiple could have the same. Theoretically, all of these could be 25%. They could all be the same as long as they add up to 100%. So we see here that the C is the highest. The C is the highest here. So that's why we see the C at the top here. Then in the next case, A is the highest. So we see that A, in terms of the probability, is the most likely to occur. So A is at the top. And then here, G is the highest. So G is at the top. So this is kind of like matching our intuition, what we would kind of expect to see. But another thing I want you to see here, guys, is that whenever we don't have much information about the majority base, that's my little logo, then the information content is a lot smaller. So in this case, the majority base is 37.3% chance of occurring, which is a C. But the runner-up G is 33.7%. So it's a little bit closer. So this is C on top followed by G. I know it's kind of squished and not so easy to see. But what you can see is that it can kind of be between these two pretty, frequently. But in this case, the majority base of A has a 53.3% chance, which is a little bit higher than for the C here. So it's quite a little bit higher here. So what this means is that basically we say that more than half the time we would expect to see an A in this position. And the rest are kind of comparable. So because if we go with A, we have a little bit more certainty if we go with the majority base of A here, than if we go with the majority base of C here, because C is very close to G. It could be pretty split between these two almost. But here, this majority is a lot higher. So we have more information because we have more knowledge about, in a way, this majority base. We're kind of more certain. If we went with A, we'd have 53.3% chance, we believe, of having an A here versus just a 37.3% chance of a C. And here, there's even more information about the majority base. It happens 58.4% of the time, so it's higher than for A in the second position or C here. And also we look relative at the other bases. Their probabilities are also quite a lot smaller. So we are a little bit more certain about G. You know, it's almost like the idea, guys, is that, hey, Yankee saying, hey, if we had to choose even one base, how confident are we if we choose the majority base? How confident are we that we made the right decision in a way that, you know, our majority base will show up? How much information do we have? Because we could say, oh, it's a coin flip. Each has a 25% chance of occurring, you know, 25% chance A, G, C, T, but that's not completely helpful. It's like saying a fifth coin flip. But if we have more information on any base relative to the others but happening, then that's pretty helpful. Then we have more information there. But I talk about more in other videos, more about the intuition for interpreting sequence logos. But here I just want to motivate that this is what it looks like. But how did I create this here? Well, let me take you to the package in R. seqLogo. The seqLogo package in R is basically sequence logos for DNA sequence alignments. So it takes the position weight matrix of a DNA sequence motif and it plots the corresponding sequence logo. So it was introduced by Schneider and Stephens in 1990. This is how you download it. So you need to install BioC Manager if you don't already have it. And then you need to install seqLogo if you don't have it. And then when you want to load it in, you just do library seqLogo. What I want us to do is use the seqLogo package. If you don't have this seqLogo package, then please install it using BioC Manager install. And if you don't even have BioC Manager, but don't worry, I have a video on how to install BioC Manager and get started with that, you know, to use Bioconductor packages. But this is how you would install seqLogo. But now that I have it, I'm just going to load it in here. Ah, that explains. There should be a 16 one. Eeks, my copying is bad. Now let's see, this will work. I have to make it a matrix so that I can use seqLogo. So seqLogo basically needs a matrix of A, C, G, T. We need to represent this same information in this PWM DF. For A, all I'm going to do is I'm going to focus on the A parts, which are 16.1 and 53.5 and 15%. So this is what I put in for A. And then for C, A, I'm going to say 16.1%, 0.161, 0.533, and 0.15. So each of the columns, I'm representing it as a row, and then I'll stitch them together. This is the probabilities for A. So if I look into A here, 16.1%, 0.161. Then 53.3% chance in the second position of the motif, 0.533. So again, we're only looking at the motif positions, not at the backgrounds. And then 0.15 is 15% for the third position. And similarly, we do this for the C part. So this is C here. So C, we have 37.3% chance of having a C in the first position, 15.5% chance of having a C in the second position, and 13.4% chance of C in the last position. So that's what this is here. So now let's look at this. Let's look at this G. So this G is similarly 33.7% chance of having a G in the first position of the motif. That's what this is saying, the first entry. 17.8% chance of a G in the second position of the motif, and a 58.4% chance of a G in the third position of the motif. That's what this is here. And last but not least, for the T, we have a 12.9% chance of a T in the first position of the motif, a 13.4% chance of a T in the second position of the motif, and a 13.1% chance of a T in the last position of the motif. So I have these four, A, C, G, T. These are the numeric. If you were to look at this part here, this matrix should kind of line up, PWM, position weighted matrix, should line up with what we have here. And again, what we're trying to basically represent here are the different positions. So this is position 1.
K equals 1, position 2 is this column here. And then this is position 3 here.
Of the motif. But we just want to stitch it in, ACGT, ACGT, 1, 2, 3, and all the represented values. So then the next thing we do is we just row bind it. That's what R bind means, row bind it together. We take each of these vectors and we just put them together. That's what PWMDF is. Looks exactly like what we were hoped it would look like. I could do class of PWMDF, and it should be a matrix or an array. That's really key if you want to check the type. So you need to have ACGT, and then you need to have this one corresponds to that one there. This one needs to correspond to this one, this two needs to correspond to this two, and this three needs to correspond to this three here.
So just to make this a little bit clearer, this part here needs to correspond to this part here. And in fact, the indexing is kind of, comparable to what you hope. This part here, corresponds to this part here. And
this part here corresponds to this part here. So that's what you need to ensure. So Yankee saying, please do your due diligence and check. Otherwise, he doesn't want to see if you've made any typos like me. Yankee won't want to see it then. Oh, he's a very lazy guy. Anyways, so we have this PWM and we're going to put it through this here. And now we have this matrix, and it looked like I had made a tiny mistake before, but it's all good because we're doing this together. So then when you put this in the seqLogo, PWMDF, we are going to get the sequence logo that we use. And this, I'm going to just copy it. I can save this image too. I can stretch it out a bit as needed, but you need for them to sum up to 1. And don't worry if you don't have them summing up to 1, or if you make a little typo, because this little software is going to tell you that you made a mistake and they're not summing up to 1. So like, for instance, like, let's just say that I put this, like before I had made this 151. So they weren't summing up at all either. And then it complained. It said that, oh, this is 0.99, this is 1, and this is 0.99. Like if I took the sum of PWMDF, then they complained that this does not add up to 1 if I change this to like 0.151, which is what I had before. So if I did like sum of PWMDF, and then I take the first column, it was like 0.99. Because again, for a data frame or a matrix, it's always like data frame, and then brackets, rows, comma, column arguments, like a matrix. So that's how you interpret it. So I'm going to fix this to 0.161, which is what it is, run it through again. And we see the sequence logo as it should be. So that's an example of basically, this is the input that we given. It's a matrix. The class is a matrix or array. So we feed it in this way here. And then we do the seqLogo, PWMDF. They talk about a lot of different examples here. And we were looking so far at, getting sequence logos for information content. That's usually what's primary. That's a default. So we can also, we can see the information content where it's basically a sequence logo with column heights that are proportional to the information content that is given. So where there's more uncertainty, the column heights would be smaller. Or we can view it this way. So we can see that in these, they're between C's and G's here. C's and G's, like a CPG island almost in a way. But here in position 5, it's a little bit lower because it's kind of being split among all 4 bases in some way, right? You can see that this is kind of lower here because it involves all 4 bases. This is slightly higher. It involves 3 bases and not P here. So there are more things we could do, but I wanted to kind of show you that we looked at a few examples again in seqLogo. We loaded these in and we saw that this was the seqLogo here. And if this is like not working, I think it's better for you to close out the session and then load it back in. So let's try to load in a fresh R session. And now you see that it's working. So basically, sometimes you need to like, just do that for it to work. And we can see that by default, what the default here is saying is that the information content is by default is set to be true. So that is what this typically is here. And they're showing an example of a probability weighted matrix, that is 8 digits long, 8 bases long. You can see that these are the 8 bases here, the example here under the vignettes, which I really like. And we can look for in our cases as well. But if we look at the information content, by default, IC.scale is true. So it's showing information content. If we do IC scale equals false, it will show the probability. So if you want to see the probabilities instead, this is the information content here. I can just do for this situation here, for this first example, I can just do seqLogo, PWM df, and then IC.scale. You can see the default was set to be true, and I can set it to be false. And I can also change the arguments like font size and y-axis and other things. But if I change this to false, we can see that probabilities are kind of mapping and matching out here. At the top, we still have like the highest one, which is the C here, then the G, then the T, then the A, and we can see that they all are at 1 here. It's just a different way of mapping. So the C's probability, G's, A's, and T's here, and then we can see that this represents the same probability weighted matrix on this scale here. But often what's more helpful, typically, and what is traditionally used, is having this information content, but you can represent it as probability. So this represents the probability on the y-axis. Whereas, and I talked about this in another video, like how this shows the information content. So columns heights are proportional to the information content, which is, you can derive them from the probability weighted matrix, but by default, IC.scale is true by default. So by default, it'll show this to you. But if you want to plot the probabilities, you can plot them like this. You see information content here, right? Like this is the default for any probability weighted matrix. And if they do IC.scale equals false, you can see the probabilities there. So it's a sequence logo with uniform column heights because they all need to sum to 1. And here, the heights are proportional to how much information content we have about the distribution of the DNA bases in a particular position of the motif. And there are other arguments you can do as well. Whenever IC scale is false, we see that this is what it looks like. And if we're using the alphabet equals RNA, then it's going to show U instead of the uracil instead of T for thymine. I just want to show you a couple more examples of how we do this. Yes, Yankee? Aww. Yankee wants to tell me to do another example, huh? You do? Okay. So Yankee is saying now, so Yankee is saying now let's bring on example 2. Example 2. Okay, Yankee, let's get started on that. So the next example we have is a little bit more, but again, it kind of does show, just summarizing the first example again, C on the top, A and G, which is kind of what we expect here. Most likely for C, most likely for an A in the second position, most likely for a G in the next position, and more certainty here. So in this next example, which I talk about a lot more in other videos, but so in the next example, which I talk about more in other videos, we talk about example 2. Again, how we use the meme algorithm for motif discovery, expectation maximization. So we have 50 input DNA sequences. Each of these sequences is 20 bases long. We are looking for a motif width of 6. That's what we specify. So we given these 20 base sequences, we get 50 of them in. We're saying we want to look for a motif of width 6. It could be any number between 1 and 20. We choose 6. Then when we ran expectation maximization algorithm, what we got is the following. When we run this to convergence, expectation maximization, because it's a process of expectation, then maximization, and expectation, and maximization. And then you run. You can think of this expectation maximization as being like, this is the expectations. Then there's the maximization, then expectation, then maximization, then expectation. And then you just running, then till we converge. When we ran it to convergence, we have this probability weighted matrix or position weighted matrix, but we also have the background here, which we don't want here. We want to just get rid of the background because right now we're trying to plot the sequence logo, so we don't want that. And this again gives us the probability of a given base in a particular position of the motif. And these again sum up to 100%. So here, just kind of, eyeballing what this looks like a bit. We see that this is like a maximum here. Is a C, 40.5% chance of C in the first position of the motif, 51.6% chance of A in the second position of the motif. We're really confident about P in the third position of the motif and about C in the fourth position of the motif. And then C in the fifth position of the motif is like 93.9%. And then G here is 53.1%. So basically, what you're seeing here is you're seeing that these are the majority probabilities. And we're going to see that since we were looking at a motif of width 6, there are 6 positions in this matrix. So this is corresponding to each and every single given position in the motif based on the width that we specify. This is always going to be for our particular width.
It's going up to width equals 6.
I just chop off this first background column, and thus the matrix that we're looking at. Yes, Yankee? The matrix that we're looking at is then these positions in the motif, this part right here. First, second, third, fourth, fifth, and sixth. This is what we will look at, and these sum up to 100%. I'm going to put this position weighted matrix into R. So let's find this. So in example 2, I basically just like copied and pasted this right here directly into here. So I thought I would do this part with you guys live. 13.8, 51.6, 2.2, 2, 10.5, all of these here, each row. So what I'm going to do is I'm just going to say A equals, and then I'm going to have this as a C here.
I'm going to just create each row here, C equals. C here is created as a vector. That's all we're doing. That's all we're doing. And G here, it needs to have these columns here. And then P here. Manoim is kind of hugging Yankee. Yankee wants to help. Aww. Aww. It's okay. I got this, Yankee. Aww. Yankee wants to help.
Basically, just try to put them in R. That's what we're going to try to do. So Yankee, do you want to help me? So Yankee's going to be helping a bit. He wants to help. So we have to do this quickly because we can't really have percent signs here. So this will be 0.3180. So I'm just converting them to decimal places really quickly. You know, this would be like 0.5160, this and comma 0.0220. Just have to make sure you put them in, like you convert them as needed. 0.02 is 2%. Oh, that was a bit of work, but we have successfully, yay, Yankee saying good job. Thanks, Yankee. Give me a high five. So we've successfully copied over this second probability weighted matrix, a bit longer. So it's double, it actually almost like double. You know, we were looking at 3 motif width before, now we're looking at 6. So now we're looking at doubling it, and we're putting this again into R now. So again, you know, this is A, it's mapping exactly as it should to the first row of this position weight matrix here, you know, and 13.8%, 51.6, 2.2%, 2%, 2%, 2%, and 10.5%, for instance. So let me just read them all in. You can see here that as expected, the A, C, G, T, these values here map to these values here now. And we're going to update our probability weighted matrix. Before it was 4 rows, you know, A, C, G, T, which is what it should be, by 3 columns for the width. Right. Before it is 4 rows by 3 columns, you know, A, C, G, T is 4, always going to be the case that this is 4. And then 3, this is the motif width. So it's going to be 4 by the motif width, basically. So now when I put this in here, because it's 6, it should be saying 1 to 4 and 1 to 6. Ah, looks like I made a tiny mistake. Now let's see where that mistake is. Ah, the mistake is here. There, okay, 1. Okay, we could have checked it here actually, because A had an extra item in it. You see, A had 7 items, so it should have been 6. You put an extra one. You guys have to be careful. So, you know, so it should be like this. Now we're just going to redo this. And now we're going to see that this is 1 to 4 and 1 by 6 here. And now this is our probability weighted matrix here. So it has A, C, G, T, and it has these 6 positions that correspond to these 6 positions here. This is exactly what we want. And again, we can check the class again of PWMDF. If in case these help you, you can check the class. You can even do the structure. STR is for structure. In Python, STR stands for string. So you always have to keep the languages straight. So this is like dim names, list of 2, character, ACGT, and null for the others, because I don't have any column names here. Like I don't have any column names here. This 1 corresponds to K is 1, 2 is K is 2, you know, 51.6, 2%, 1.9%, 44.5%. So now we could do the seqLogo on this. Aww. Aww. Yankey's being very affectionate right now. So Yankey's excited because now we can view these results. He wants to close his eyes till I guarantee that the logo comes out right. Let me check. Actually, Yankee, I think it's fine. You can open your eyes and see. So let's see what a beautiful logo we hopefully will have. So this is the seqLogo PWMDF, and it generates something like this for us, which is really cute. Information content, again, this is in bits, B-I-T-S, bits. Maximum is 2. When we are at the height of 2, we have the most information. That means like we're very certain. Like it's pretty much like we're pretty certain that it's only going to be 1 DNA base, for instance. That's a maximum thing. Like maybe it's like a 99% chance of having a given DNA base. So the closer we are to 2, the more certain we are and the better information we have. So it's about how much information you have. So let's copy this guy here. So as expected, if we look at the tall ones here, the majority like we were looking at before, so C is on the top here, then A, then so C is at the top here, then A is kind of tall, followed by T below, but A is on the top. So the whatever is on the top is usually the one that has the highest probability, or it's tied with the next one below it, for instance. And we'll look at that soon. So C is on the top, followed by T. Less information than A here, and followed by T. The majority has slightly more, you know, A is a majority, and 51%, 0.6, which is more than 40.5%. So that's why it's up here. And then this T is 91.9%, so it's up here. And then this C here is at 93.9% and 93.9. So these are the same. So these are the same here. 93.9%, 93.9, these are the same. Slightly better than this situation. If you had to look at the height here, it's comparable, but this is slightly higher. These are the same height of 93.9, 93.9, and this is 91.9 here, which it increases because, you know, for these C's, because the majority is higher slightly. So even more confidence about C here. And these are the same because of this here. And we lose information because, when we get down here, because this is G and T. So, it's, you know, we lose information here. But the key idea is that when you're looking at this probability, sometimes things may not stand out so clearly for you. So the key thing is that when you're looking at these probabilities, sometimes things may not stand out to you as clearly. So the point is of this sequence logo is to kind of tell you how certain are you. Like if you were to check out these probabilities right here, you would say, oh man, this looks like it's really high chance of a T and then a C and a C. It's like so close to 100% that you can pretty much reliably say that out of like 100 sequences that you look at, around almost 91 to 92 sequences would have a T, you know, on average in the third position of the motif. And similarly, you could say, you know, between 93 and 94% of the sequences, you know, out of 100, 93 to 94 of those sequences could have a C in the fourth position. That's like pretty high. So you are really confident about having a T and a C and a C here. That's why it's so high up here. You're not so confident about the situation. You can have a C or a T here, but there's still a 7.4% chance of a G. You know, it's kind of there, and then an A here. But the other probabilities here are so low that we, you know, they're getting squished to the bottom here. So that's how you can interpret it. Like, so this really shows you that we're really confident that in this position, it's a T, then a C, and then a C. And I have another video just talking about how you can calculate this information content from a probability weighted matrix. So please do see that. How do you calculate these heights, which are then used to stack and plot these DNA base are called the residues. So I have a video that talks about how we can plot this, how we can go around and figure this out, basically, to do this conversion. And it is also in the bio here. Third example that I want to show you is the Goldilocks example. So it's just in the middle. And this is an example that hopefully you may remember. It's from Gibbs sampling, which along with the expectation maximization mean approach, Gibbs sampling is another approach that we can use, Yankee, to do motif discovery. It's another such approach that's out there, huh? It's another approach that we can use. So let's look, and I'm going to show you a probability weighted matrix that we end up learning in the examples I go through this, where we are running the Gibbs sampling algorithm for motif finding on a set of n equals 10 input sequences with a motif width of 4 nucleotides. And we are seeing the current state of the Gibbs sampling. Current motif occurrences that we have found are given by these red underlined parts. So these are like the 4 base motifs that we are seeing. So it's like in the middle. It's not as short as 4 sequences, 6 letters wide, 3 base motif. It's not like that, but neither is it as big as looking at 50 sequences of 20 base long sequences of sequence lengths and looking for a motif of 6, which was the second example. So now we're going to look at 10 sequences here. But with Gibbs sampling, we slice off 1 and we focus on the other 9. So we're kind of looking in a way at 9 sequences, but looking at a motif width of 4. So please suppose that in the next iteration of the sampler, sequence 5 is picked and the motif position in that sequence is to be resampled. So please note that we use a pseudo count of 1 for our estimates. So we assume a standard probability weighted matrix model with no prior information being used. I have videos on palindromic model as well. So we compute the parameter estimates P that are calculated in the predictive update step, given that sequence 5 has been selected. So I talk about how, if you select any sequence, like here if it's sequence 5, we just cut out sequence 5. So 1, 2, 3, 4, 6, 7, 8, 9, 10. We cut out sequence 5 and use these 9 sequences 1 to 4 and 6 to 10 here. So please again do subscribe to my video to see how we can do this Gibbs sampling algorithm and land up with this, position weighted matrix, this probability weighted matrix. And again, as before, we are not going to look at the background. We just wanted to look at the motif parts. Yay. So we're going to focus on just these probabilities. And as I was mentioning before, here C and P, for instance, they are sharing the same probability of occurring. So it really depends on how if we use different approaches, like seqLogo can decide maybe to put C on top or P on top, So that's what I meant, that sometimes the majority can tie. And here they have the same again, but there's a majority at A, so A is on the top. So C or T could be on the top. So if a base is on the top, it either has the highest probability or it could be tied. So you have to just double check and see the relative heights. The height should be the same in each column, then they have the same probability. So the height is kind of telling you in a way the scaling. So the height is telling you, that T is way more than C and A. But here we can see that, C and then is the highest here, but then T, it kind of looks really the same because it's just off by 2% or so. But so C is slightly taller than T, and which is quite a bit taller than A, and G is very negligible. G is smaller. G is around like half the height of A, so to speak. So in any case, we can take this here and similar to what we did before, we can just put this in the seqLogo. So let's do that. For this example 3, I just put in these probabilities that we've been working with for this example 3 here. So basically, this means a 15.38% chance of an A being in the first position of the motif. It's 4 bases long, is the width that we were looking for. And then a 46.15% chance of an A being in the second position of the motif. And again, the 4 bases because the motifs that we were given here were all 4 bases long. 4. The rest are just background. And we talk about in other videos like how we can estimate all this. So, then there's a 7.69% chance of an A being in the third position, 15.38% chance of an A being in the last position, the fourth and last position of the motif. So again, this is always like what the width is of the motif.
So this is equal to W.
So this is our A here. Then we can also see what let's just load them in C, G, and P. Like for instance, let's look at G. 23.8% chance of a G in the first position of the motif, 15.38% chance of a G in the second position of the motif, 61.54% chance of a G in the third and last third position of the motif, and a 23.8% chance of a G in the fourth and last position of the motif. So these are the variables here. So what we're going to do is we can see that these all, 1 to 4, yep, as expected, these map to these here. So we can just kind of load this in this probability weighted matrix. And again, it has to be a matrix. A, C, G, T are the column, are the row names here. And the column names don't have anything. This one corresponds, the first column is the first position. Second column is the second position. Third column is the third position. And fourth column is the fourth position. So just to hit that home a bit more.
This column.
It's like a rainbow. So yeah, they're the same. Great. So then the next thing we do is we just do seqLogo. And we will get this new one here. And again, this PWM is 1 by 4, but 1 by 4, it's always 4 rows. So it's always going to be 1 to 4 is the ACGT, and then this is 1 to W width. So this is the corresponding sequence logo here.
That we get. We can copy it to clipboard and scale it as needed.
But you can see again another example of this probability weighted matrix becoming a sequence logo. So the interpretation for this is that if you look at the majority section here, then 46.15 is a C here, that's for the C's here. Then 46.15 again, A, and everything else is pretty much the same, 46.15, 46.15, 15.38, 15.38, 23.08. So it's all the same proportion of all the characters here. So even if these were the same, the heights could be different based on like what this would be versus what this would be, for instance. So it's important to note that, even if the heights are the same. And we can maybe play around with that and see what we could get, for instance. So if these were the same and all of these are the same, for instance, then the heights are exactly the same here. And then in this situation, G is really much higher. If you are viewing it relative to the other one, this is 61.54. And here, these are kind of tied here. So the key thing is here, we're 46.15% confident about the majority base being in this position, C in the first position. Same for A being in the second position of the motif. And then here, 61.54% chance. My little guy. 61.54% chance of a G being in the third position of the motif, which is a lot higher than 46.15%. So this is quite a bit higher because the other ones are shrunk down to 15 and 15 and 7. So we have more information here in this base than we do in the second and third, like 61% chance, 61.5% chance of a G being here. It's quite a lot higher than a 46% chance, right? But this base is in a poorer state because it can be between a C and a T. We can't even say a majority. So it really depends. In this situation, what they put on the top, I think, was the T's in red. Then they put the blue, which is a C. So it's so small here because, they put T and then C. It can be either or on the top. It could have been a C and then a T, then a G and then an A. These heights should be the same here relative, but I'm just like trying to keep it consistent with the height. So it could be like CTGA or TCGA. Oh, the Cancer Genome Atlas, that's actually like a data resource in bioinformatics, but they chose this one here. So kind of motivating to you, another quick thing, while we're at it, is that like, what if instead, the majority is still the same, but like, what if we just kind of tweak this a bit? Where the majority is the best base pair. Let me highlight this in yellow. Oh, wait, not that one. What am I doing? This here, and then this here, for instance. So I said these are the same and all of them values are the same, heights are the same, but let's say that instead we bump this guy up to being 20. So add 5% more and let's subtract 5% from here. And let me show you what happens, even if they're the same height, if the next one, if it looks like this, for instance. So we can go back to R and just see what that would look like. So I'm just going to make this, the change that we made is in the second part here, these two rows. Okay, so let's just change those two here. So let's bump this one up to being a 20. And let's make this a 10. We've added 5% to this one and we subtracted 5% from the original here. So now let's see what will happen. Okay. So we can kind of see that in this situation, that this one is kind of higher than this one. You can sort of see that we went from, in this situation where they are the same, here 46, 46, 15, 15, 15, 15, 23.08 here, that now we kind of have more like, three majority. So this one is kind of really tiny. So at least we can say that G is really small chance of happening. Whereas here they were kind of all like kind of average, close to 23% or 15% and 46. But here we made one much smaller and said that we can kind of rule it to these three more. This is 46.15% and this too. But that, these are 23.08% too. So these are kind of like the same as well. But what we are seeing is that we definitely, like we could be ambivalent, like we could say, oh, it could be an A next or a T next, but in this way, why this is better is that we can say that there's A, then comes T, then comes C and G. So there's more of an ordering. So there's more information here. But here we're like, it can be a C, then a G, then like it could be a T or an A. Like we don't really know. That's kind of why this is really key here. And to motivate that again, like, just to see that even if we have these being the same and these being the same, the key difference is that here it can be between the third place is tied. But here we can at least say that G has the smallest probability of occurring. We don't really want ties. Like this was what corresponded to the lowest result because there was a tie between C and T as well. We really just want to choose one majority. We want to be able to say it's going to be this base most of the time. So if it's tying, that's usually not really that good. But it's still better than 25, 25, 25%. In fact, like I can also show you like what would happen if we had like, a fifth column in this and that happened to be like 25% for each of them. But what would happen? Like if I were to do this. So if I were to do this, just make them all 25% just for reference. Then what this would become is I could add in, like, let me just add one more column for each of them, 25%. This is like, the typical, like what we would expect, just by randomly, just choosing a base. So we added in this fifth column of 25, 25, 25%. So you can see that we added that in there. And now we can do the seqLogo here. And we can copy this image here and kind of see what this looks like here. You can't even see it. There's virtually no information from this. You see that whereas before there were 4 positions, now there is a 5th position here. And even if I made this like 26, for instance, and I scale this down to like 24, let's see if we get anything at all from there. Probably not. If I made this a 26, so that it all sums up to 1, otherwise it'll complain. Now let's see what we get. Still not marginally high enough to detect anything. So we can keep on playing with this. Maybe let me just make this like 30. And then let me make this like a 20. And now let's see what we get. Will we get anything at all? Yes, we get something really tiny. Something really, really tiny. So I encourage you guys to take advantage of this tool here and then to just, play around with this now and see what you get. Like we can say that this is like 30, for instance, and then we round this up to 20. So they all still sum up to 100. And this is kind of tiny, but we do get something, but there's very little information because the only thing we can say is that, A, maybe more than average, C, definitely less than average, and G and T, what you typically expect. So maybe a little more of A's, little few C's, and, kind of roughly same amount of D's and T's as you would expect. But that's all we can say. There's very little information here. There are various data resources out there. So we can see that I've had this giant object here that I was looking at. And the way that we can look at it is we can see for different types of, for instance, transcription factors like ZBTB33. Like, let's look this up here. So this name of this motif is Homo sapiens encode motifs ZBTB33 BISC3. So let's look this up here in this list that I have.
So if I look this up in this list that I have here,
I created this list object here. And what I have seen here is I can see the strands and the counts here, but the background probabilities that we usually assume like 25%, 25%, 25%, that may not always be the case. And we've looked here at like, at backgrounds that are not 25%, but we just kind of, sometimes we can assume this as a blanket statement here. And then we can have a position weighted matrix, and these all sum up to 1. And so I can look at this matrix here and then see, okay, this is what this matrix looks like for this given transcription factor. This is what this looks like. So another thing I kind of want to show you is that a lot of times we can see that that there are different probability weighted matrices for different transcription factors. So I just wanted you to see that, that we can even look at like a probability weighted matrix for a transcription factor like REST, which could be very key here. So this is data resource that is called motifsdb. It's also available on Bioconductor, so you'd have to install BioC Manager ..install motifdb here. But this is kind of interesting here, but there's also this motif database that we can look at here. Maybe I also want to look up like SPI1, for instance. And there are 4 motifs that I can identify for that. Or maybe I want to look up like REST, for instance. Let's look up this transcription factor REST. So there are 3 motifs here, and I can look up what motifs look like. They are found from Jasper database. There are a few different ones. These are the 3 that were found. These are the IDs that were found for these. So one thing I can do is I can already, so if I do motifs 1, this will give me, it will say that we are getting this from Jasper 2018 for Homo sapiens. It's going to take me to this basically. So this is a data resource here. And I can already say, okay, so if I look at motifs 1, this is going to give me this top one here, which is going to be from here. And this is the probability weighted matrix for each of these positions. 1, 2, 3, 4, 5, 6. You can see in some positions, we are so certain that it's that base. So in the 4th position of this motif, we are super certain it's a C, for instance. And then in others, we may not know at all, but we know it's definitely not certain bases, which is always great information to know. So this motifs 1 gives us the probability weighted matrix. For this, for instance, for this REST transcription factor. So we can also just kind of put this in. So we can just do PWM1, for instance. So we see this here, and then we can always just do seqLogo. but we have to basically make this into a matrix. We looked at a few examples again with seqLogo. We loaded these in and we saw that this was the seqLogo here. And if this is like not working, I think it's better for you to close out the session and then load it back in. So let's try to load in a fresh R session. And now you see that it's working. So basically, sometimes you need to do that for it to work. You can already just kind of query this and see that there are different types that are available here. You know, like we can already see that if we look at motifs, we have like these 3 different motifs that are available for REST, for instance, a transcription factor. In Homo sapiens, we can look at different data resources here. And then this PWM motifs 1 will give us a probability weighted matrix for the first one, because if we look at motifs 1, it's an object here. Motifs 1 is going to tell us about the first motif, which is this H sapiens Jasper rest, this thing right here. Will be what this thing is here. And this is the respective probability weighted matrix for A, C, G, T, the different bases here. We can look at motifs 2, for instance. But before we do that, you can already see that in motifs 1, there are some really great positions, like the 4th position where 100% chance that it's going to be C. That is awesome. That's what we like to see. Like, and some that are flat out 0, that can never happen, which is also really great. And there's some areas where we may not know as much about, or there may be some uncertainty. Like here, maybe this first base not the greatest, or the second base, but there's some that it's really, really good, strong results for, like 4-1 as well, or even like the 14th. So, you know, that's the probability weighted matrix there. This is for the second one here, you know, corresponding to this here, 0.2. And we can do PWM2, for instance, and then we can extract what this object is. You can see that this one is, instead of being 19 bases wide, the motif is 21. You can see that in the real world, these kind of become like a little bit bigger than we expect. And this motif that it's binding to can be kind of different. But 16th position looks kind of good, like really a strong probability of 98.3750% chance of an A being in the 16th position. And then in the 3rd and last position, we can also see this too. So this is 22. And, you know, this is a good position. 7th position, 94.8% chance of an A being in the 7th position of the motif. But here the width is 22. And again, all the columns sum to 1. So if I took like sum of, let's say, PWM 3, if I just wanted to sum the 8th column, for instance, it would be 1. 8th column would be this. I had shown an example for REST as well, for REST transcription factor. Let me kind of show you another example, like NFKB1, for instance.
Like it's just loading in motifdb, this database.
So there are 4 motifs here that show up. The 4 motifs that we find here are mouse musculus, Homo sapiens, Homo sapiens, Homo sapiens. So I want to just kind of look at maybe motifs, and then maybe I can look at the second one, which is over here, Homo sapiens, Jasper. So this kind of shows you that there are 2 organisms if we look at motifs. One is a human, and then the other one is a mouse musculus and human, and then a conilus as well. So we can look at motifs too. So I can look at motifs and then look at 2 NFKBs are really key transcription factors. So we're super certain that the first base is, and the motif is a G, G, G. It starts off as a G, G, G. This is what this matrix looks like here. It's a probability weighted matrix. So I can just call it like PWM2, and I can just say it's motifs 2 here. And then what I can do is I can just do seqLogo PWM 2. And yay, indeed we have this here. We can see that this is the information content for one of the motifs. The motif here that it's binding to is, this is the motif, this Homo sapiens Jasper NFKB1 MA0105.2. This is this information on where the sequence logo for NFKB1, the second motif that was found by this Jasper database. And if I look up this information, this ID that we were seeing there, if I look this up here, you know, Jasper is an open access database of curated, non-redundant transcription factor binding profiles stored as position frequency matrices, PFMs. So, excuse me, you can see the frequencies as well, information, and one can derive the other and so on. But you can see here that they even have this meme that I'm talking about, meme. Motif elicitation using expectation maximization. So this is for the second one. And again, we can also just see the probability breakdown as well. That should show you, you know, if I do ic.scale equals false, it's just going to show me the probabilities. And you can see that this is like GGG, you know, and then this is like 88% here, and then 11% is A. So that kind of makes a bit more intuitive sense here. But this is like 11% here. And this is like 88%, and A, G, C, and then all the other bases, we can show the probability weighted matrix, a position weighted matrix this way. Or we can show it with information content where the height shows us where we're the most certain, which I feel like is a little bit better to tell us which ones are we more certain about and which ones should we choose there. We choose where the information content is really high. You can get the same information from this, but you just have to convert it to this scale where you're like, not only are you showing the bases that are the most likely in a given position, but you're kind of weighing it by how much information we have, as we talked about. So like here, we're really certain it's a C, so it's like kind of like around 2 or so. And when there's less information here, like in position 5, it could be 61% chance of an A, 61.1% chance of an A in position 5, and 33.3% chance of a G in position 5. So there's a little bit more uncertainty here. It's not quite as clear cut. These are all these probabilities. We can even look at motifs 3 that we got from looking up in the motif database, looking up a particular transcription factor. So NFKB1 is a transcription factor, nuclear factor, kappa beta related factors here. NFKB1 is a pleiotropic transcription factor present in almost all cell types and is the endpoint of a series of signal transduction events. It causes inflammation. It increases the production of inflammatory cytokines, chemokines, and adhesion molecules. So it is really important in COVID-19 as well, in the cytokine storm that we see there. It also regulates the cell proliferation, cell death, the changing of the cell, and differentiation as well. It increases macrophage. These are like those little like gobblers, immune cells, and myofibroblast content during tendon healing, etc. So that's kind of what's key about it. And there's more. It helps regulate the expression of various genes as a transcription factor. NFKB1. And the gene regulatory networks related to NFKB1. You can see NFKB dependent gene transcription, right? Like, so NFKB is really important and very critical. Inflammatory effects as well that we are observing. The NFKBs, it belongs to that family, the immune system functionings of NFKB1. So it is a transcription factor that does quite a lot of things. So you have to be very careful about NFKB1. It's kind of very huge transcription factor. So we have to be careful when we're making discoveries related to it. So for motifs 3, we can also look at, this is the ID here. So it's a different ID. Whereas for motifs 2, this is the motifs 2 here, and then motifs 3. Oh, the difference is instead of .2, it's .3 here. Okay. And now what we can see is we can also just get this probability weighted matrix here for motifs 3. This is what it is. So it is 11, whereas before for the second one, it was also 11. The third one is also 11 here, but it'll be kind of different, which is what we'll see here. So we can make this PWM 3 here, and 3, and 3. So let's see what the third one doesn't start out super strong with G and G here. So we can see what this looks like here. Okay, so G and then G here. So it's kind of a bit different, you know, this probability weighted matrix. So let me just clear this a bit so we can see that that this is a bit like, you know, it can be G here and then G, but there's a little bit more uncertainty, which is what this information content shows for NFKB. We can, you know, we see the sequence logo. We can also see the probabilities as well, and these would be summing to 1, whereas the information content is proportional to the information that we know about it at a given base. And then lastly, we can also see motifs 4, and this one will be .4 here. We can also just do the same here, where instead now this is 13. So before we were looking, the earlier two were 11 bases the motif. So this motif is 13. So it goes up to 13, the first position, second, third, fourth, fifth, you know, all the positions. What is the probability of observing a given base A, C, G, D here? So this is here, it's going to be 13 bases long. So that's the 13 one here, and we can view what this is here. This is the information content that we have here, which kind of seems like its majority more is A here, then G, 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 G here. You see how that kind of looks? But when there's more uncertainty, then it's not so high. But here it's super high because it's like we almost have a majority base, so to speak. So I just hope that you can kind of compare and see how, you know, they do with each other. And let me also show you that they're kind of like the same information, but I mean, you can go from one to the other. You can just see how they kind of map to each other in a way where if there's A, C, G, T, A is not as tall in probability as the majority like G, G here. So that's why this information is a lot less. In these cases, it's a bit more. But you know, in this position 4, there is some amount of T, which is what drives this guy down, and some amount of A here. And in position 6, we can see that there is A, and to some extent, and then there's G, but more A, but we can see that this G is relatively taller than this G here, which is kind of making it a bit more uncertain about this majority base A, which is what drives down its height here. So you can see how, you know, when there is a bit more sharing that's going on between, and one is not having all the thunder, the information content is kind of not so high because it's kind of sharing the spotlight. Like here, A is sharing some with T here. You see A is sharing some with T, so that's driving it down. So it could be either A or T, you're not as sure here. But here, this could be T and C, maybe to a lesser extent. There's another base. I think that could be a G here in position 8. It's because we have T and then C here. And that's why like this C here is a bit more than this G here. So this T is sharing a little bit more with the C. So the information content in this position is less. So it's kind of like this different ways, but we typically use this information content because it kind of gives us not just a sense of probabilities, because you can already see that kind of from a probability weighted matrix. But if you're visual, this is really helpful, but this will give you more information on what are the bases that are likely. Like it'll be like A, G, 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 A, T, C, C, C. So that's how you kind of map them here. But you're not so confident about this. You're really confident about these, which is really great. I hope that this was helpful for you guys to see how we can use seqLogo to plot the sequence logo given a probability weighted matrix. So please do reach out with any and all questions. We always have time for you. And please let us know anything that you want us to see next as well. So I'm Saniya Kuller. This cutie here is Cute Little Yankee. So if you found this helpful, please like and please subscribe, and do please see my other videos in this description below and on my channel as well to provide more context. So here is a huge hug from my cute little Yankee. Here's a hug from cute little Yankee. So here's a hug from cute little Yankee. We're at the beach. Thank you guys. Yankee's also going to be blowing kisses. Aww. Thank you guys. And again, I have other videos on talking about how to make a sequence logo online from a set of motif occurrences. There's so many tools out there. I hope that this will help you guys. Have a great day, guys. We're going to the beach and just chilling.